Hi, welcome to lecture on EnvAdapt adaptation of animals. Now we will have short review on how we are regulating the body fluids in different osmotic pressures or in different kind of environments. It depends on, are we losing water more than we are getting from somewhere else? And for that reason, we are checking on the osmotic pressure of urine versus osmotic pressure of plasma. And it might have different implications for excretion in the water excretion or on the solute excretion. So how much water? Or solute, for example, sodium or chloride or whatever. And then it can have, of course, effects on the composition of blood plasma. So, first of all, the iso-osmotic urine. So it means that, okay, now there is as much water, as much sodium, as much chloride and all other stuff in the urine as in the plasma. There might be that the ions are not the same, but there might be that we are excreting more urea than sodium or whatever, but anyway, the idea of all this summa of all these solutes is the important. And then its effects, of course, on the blood plasma. So, but because we are losing as much water, then the plasma osmotic pressure is the constant. And that's very typical in the freshwater crabs and marine invertebrates. So they are lacking the osmotic regulation. Then the second way is on the hyper-osmotic urine. So now the osmotic pressure of urea is smaller than the plasma. So now we have excreting more water and the solutes are held back. And the osmotic pressure of the plasma is raised because we are gaining these ions, etc. back. So we are excreting mainly just water. And then we might have also hyper-osmotic urine. So now we are reducing the water release, so we want to save the water, and we are using this excretion mainly to get rid of the solutes. And therefore, if we are taking the water back, then we are lowering the osmotic pressure of the plasma. And in the humans, we can regulate it very drastically. So we can have hyperosmotic urea, we can have hyperosmotic urea, etc. So we can have release mainly water or mainly solutes. And this hyperosmotic urea is not very typical. And it's linked to animals that have risk of dehydration, like we humans. Or all the warm-blooded animals that we are, when we are respirating, we are losing water much more. Okay, what about then in the food? So we are not getting water just by drinking. Also comes in the several forms in the food. First of all, when we are eating wet food, of course we can get water. But the problem might be that there is if it's a salinity. So if we are taking marine invertebrates, they have so huge amount of sodium and chloride, so actually we might lose more water than we are getting from this food. Also, we have plants called halophytes that are accumulating ions inside them. And again, many animals are not eating them because there's dehydration risk. And this is the same as the problem is that, okay, if we would drink seawater, the seawater have high chloride, and when we are getting rid of this chloride, we are losing more water than getting. But also in the food, we have metabolic byproduct. So we can get metabolically product of water, and it comes in a little bit more or less, and actually quite high numbers. So when we are eating mainly lipids, for each gram of lipid, we are getting 1 gram of water. Then in these others, maybe half gram of water, but you can see that depending on what kind of nitrogen waste we are producing, then there's a small difference in the water content we get. And this can cause that, okay, if we are eating only proteins, that can even cause the dehydration. So it's, if we are eating only some food from which we are not getting water enough, and then not shrinking, that could be also a problem. So how we can then minimize the water loss? So when we have higher metabolism, we need oxygen, and for that reason we need respiration, and therefore we have this obligatory respiration water loss. And of course, when we are getting rid of the nitrogen waste or feces, there's also water that we are losing. There's a kangaroo rat that is living in the deserts, and it's not drinking at all. It gets all the water it needs from the dry seeds. And they've been testing. Okay, kangaroo rat and lab rat. In the metabolism, they get as much water from these barley. And on the respiration, there is no difference. This urea is more concentrated in the kangaroo rat, so it's losing less water. And the interesting thing is that in the feces, it's very dry. It's actually smaller amount of water in the feces than in this barley. So that's why it's marked that, okay, it's actually 0. And then we can calculate that, okay, what is the net gain? And because this is over 1/0, it gets more water than it actually needs by just eating this barley. Whereas this lab rat, that would cause dehydration. Okay, what about then osmosis, cell volume, and dissolved entities? So we know that, okay, water is moving to higher osmotic pressure because there is less free water molecules. And if the cell is moved to hyper-osmotic environment, then of course water is moving, and that caused the swelling of the cell. But if we reduce the content of the cells, then water is not moving anymore. So now it's in now it's iso-osmotic. And of course, if the cell is moved on the hyperosmotic environment, usually water is going out. The cell is shrinking. But if we are increasing the content of dissolved entities, like having more salt there, actually we are not using salt, but anyway, we can have some molecules there that are soluble, and therefore now it's again iso-osmotic. Okay. Does this isoconformer have osmotic explosion? So when you are changing environment drastically and then having very high osmotic pressure, does it mean that okay, your cells are shrinking or just breaking down because they are explosion? Okay, let's have rising the osmotic pressure, and let's look on these yellow balls and the blue balls. So one is in inorganic ions like sodium, potassium, chloride, etc. And then these organic solid molecules. So this osmotic pressure rise, this animal is increasing the soluble particles by limiting the cell limit the cell volume changes. So it's still iso-osmotic. And these mussels and crabs are doing it by catabolizing the proteins. So you have a protein with several amino acids. And then, when you are catabolizing them, you have several free amino acids there. And then they can also transport more amino acids in the cells. And therefore, it's still iso-osmotic. So you need have more of these yellow balls there. But the amount of these blue balls is actually the same as in the lower osmotic pressure. So there's no change in the concentration of inorganic ions. And that means that, okay, if you have any electrical regulation, it still works in the same way. Okay, then how we can minimize the side effects? The problem is that when you have anything more in the cell, that might affect on different processes. And that's why they are using osmotic effectors to regulate. So something that is not regulating other things. And we have certain molecules that are having minimal side effects, like some free amino acids, then some betaines that are also amino acids that are methylated. Then there is taurine that is also happening in humans, and there is glycerol that we'll be discussing in the freeze avoidance. And in some cases, we might have also counteracting solutes. So we have first osmotic effector with a minimal side effect, but because it's affecting, then we can have another molecule to counteract it. And for example, in these elasmobranch, shark, rays, etc., and they are using urea as well as these methylamines in the body fluids. And it's been tested that, okay, when having, for example, pyruvate kinase in different urea concentration, you can see that this red line is rising. That means the inhibition of this pyruvate kinase. And just with this methylamine, trimethylamine oxidase, that will activate the enzyme. So one is increasing, one is decreasing the activation. But then when they have the mixture of these two molecules, then this enzyme is working perfectly well. Thank you.